Now we're going to talk about molecular models, which is actually kind of cool because now that we've had a lot of this background and theory about shapes and geometries, I want to show you some real examples of what actual molecules might look like if you were able to see them. Okay, so the point is that every single molecule on Earth has some kind of shape, whether it's tetrahedral or linear or trigonal planar or whatever, and these shapes are usually very, very tied or related to what they do. In other words, there's some sort of relationship between the structure and the function, okay? And one of the things that chemists and other like-minded folk try to understand is, if we know what the thing looks like, can we figure out what the thing does, right? What kind of function does it have? Okay, and so here are two examples of molecules that you might be familiar with that have these kinds of properties, okay? Over here on the left is aspirin, also called acetyl salicylic acid. And in this picture, you have different colored balls, right, that represent the different atoms, okay? And so all of the gray balls, like here in the middle, these are all carbon atoms. And all of the light-colored whitish balls are hydrogen atoms. These are all H's. And the red balls are oxygen atoms. And so this shows you kind of who's connected to whom. And you can see that the molecule has a certain shape, okay? Over here on the right is a much more complicated molecule. This is part of the 16S ribosome in bacteria. This is basically how prokaryotes like bacteria make all of the proteins in your body. And the point is that if you could zoom in, you would see thousands and thousands of little atoms that are all connected to each other in a very, very precise way. Okay, and so chemists, biologists, physicists, people like that tend to spend a lot of time trying to understand the exact 3D shapes of molecules and try to figure out these relationships between structure and function, okay? So we're not gonna really focus on really enormous things like the 16S ribosome, but what I do wanna do is spend a little bit of time telling you different ways to depict these models or draw them differently. Okay, and so the one on the left is one that you saw literally 30 seconds ago, the ball and stick model for aspirin. Okay, and so as the name implies, in the ball and stick model, all of the balls are the atoms, and all of the sticks are the bonds. So you can see here all of the different atoms, or all these different colored balls, for example, the carbon and the hydrogen. And this stick right here represents the bonds. And the way these sticks work is if you only see one stick, that's a single bond, but something like this would be a double bond. Okay, something like this in the middle, this is actually resonance. Okay, so the thing in the middle is actually a benzene ring, which looks like it should be alternating single and double bonds. But actually what happens is you have something called resonance, where the extra pairs of electrons are being spread out or delocalized amongst the whole thing. And so these dotted lines try to illustrate that those electrons are moving around. On the right side is something called the space-filling model. And so this is meant to give you a slightly more accurate chemical version because all of the sizes here correspond to atomic radii, also known as van der Waals radii. And the point is that larger atoms would have larger radii and you would see them bigger. Okay, so for example, here's carbon in the middle, here are the hydrogens on the outside. Even though this might be a little bit more accurate chemically, it's a little bit harder to see in terms of what's going on and what kinds of bonds you have. And so unless the molecules are very, very small and have a relatively small number of atoms in them, people tend to stay away from this space-filling model. But it is useful. So for example here, this group down here and this group up here in the ball and stick model look like they're kind of far away, but in the space-filling model, you can see that they're actually pretty close together, and these are actually touching, or almost touching. Now for really, really, really big molecules, okay, like the ones that you find in biology, like the 16S ribosome, you might have something called a wireframe model, where we don't even show bonds anymore, or sorry, we don't even show atoms anymore, but we just show the bonds, so the sticks of the ball and stick. So bonds only. And even if you only show the bonds, you still have lots and lots and lots and lots and lots of bonds in there. And so it gets very cluttered. And so a simpler way to do it is to show cartoons, okay? Literally like cartoons, okay? These are not meant to be very accurate, but they're kind of easy to see, and they're pleasant to look at, okay? And so the way that this cartoon works is I color-coded this, so everything that is made out of RNA is in gray, and everything that's made out of protein is either pink or yellow. And if you zoom in, you would see that all of these gray areas have certain types of helices, which just kind of coil around and wrap around each other. And then in the protein, all of the pink parts represent what are called alpha helices, which also coil and wrap around each other. And all of the yellow parts represent things that are called beta sheets, or beta pleated sheets. So these are just different ways that these atoms can arrange themselves in 3D space in these enormous molecules.